Have you been to the microlab? In this video, we're going to look at some of the equipment that's needed for doing experiments in microbiology. But first, what do you remember from your last lab? Let's just try a warm-up question. PPE, or personal protective equipment, is essential to performing laboratory investigations safely. What is the order to don PPE? Remember, don means to put on. Pause the video, come up with your answer, and then join me when you're ready. PPE is essential to performing lab experiments safely. After you wash your hands, you're going to put on your lab coat. Then, if you brought a mask with you, you'd put your mask on. Next, you'll put on your safety glasses or goggles, and then your gloves. Did you come up with that in order? Some of you listening to my voice are already familiar with how to put on PPE, but not everyone will be, so I've got a little song to help you remember. Feel free to sing along. In the lab wear PPE, it means safety for you and me. Wash your hands with lots of soap, don your lab coat and button it closed, respirator and check its seal, goggle and gloves complete the deal. So your gloves are always going to be the last thing that you put on. Then, after you do your experiment, once you're done, it's time to doff, tent and pull and hook gloves off. Remove your goggles and store away. Bag your coat for another day. Doff your mask, don't touch the front. Scrub your hands and now you're done. So once you're ready to leave the lab, it's time to take all of your equipment off. Your gloves come off first. Remember, you're going to take the thumb and forefinger of the hand that you write with. You're going to use that to pinch up the glove on your opposite hand. Then you're going to pull the glove, that little tint that you make, towards your fingers to actually get the glove off. Then you're going to use the pinky of that degloved hand to slip it under the cuff of your remaining glove, and then you're going to lift that and roll it over your hand, forming a little packet between your gloves that can go in the trash. Next, you'll use your hands to remove your goggles, put them away, and then in micro, we always keep our lab coats in little Ziploc bags, and we'll keep our pens and pencils there as well. So you're going to fold your coat up, put it in the bag, then squeeze all of the air out and seal the bag for another day. Next, you're going to take your mask off. So if you're wearing a mask with ear loops, you're going to use the pinkies of both hands to hook them under the ear loops and remove your mask without touching the front of your mask. And then you're going to throw your mask away or store it. Then you're going to wash your hands. So that's what you would do under normal circumstances when you're going into a patient's room or you have to visit someone who is under quarantine. Mask is always the last thing to come off, and you only take it off when you're in a safe environment. So that's cool and everything, but why do we wear PPE? Well, PPE is part of what's called the aseptic technique that we use in microbiology. Aseptic technique is basically the set of steps or procedures that are taken to prevent contamination of your person, your environment, and your experiments with unwanted microorganisms. And your aseptic technique actually starts when you come to the lab. Right after you put your book bag and your coat and whatever else you brought away, then you'll go immediately to the sink to wash your hands. And this is going to help wash off some of the microbes that you might have brought on your hands into the lab. Next, you're going to wash your desk with 70% isopropyl alcohol. That's going to kill or remove any microbes that have settled there or been left prior to your arrival in lab. Then you go back to the sink and wash your hands again just in case you got microbes on them. Next, you're going to put your PPE on as we described on the last two slides. Okay. Then when you complete your work, you're going to do basically the same things in reverse. You're going to wash your desk. Then you're going to start removing your PPE, and you're going to wash your hands before you leave the lab. This way, you're not going to bring microbes from the lab with you as you leave the room. One of the most important tasks that happens in any micro lab is growing microbes. But how do you do that? Well, if we have any hope of success, we have to provide the factors and conditions that microbes need for life and reproduction. What are some things that microbes need to have in order to grow? Pause the video for a few seconds and make a list. Okay, let's see what you have. I see you have some things here. The first three things that microbes need are water, nutrients, and proper pH. Water is essential to all life. It helps essential chemical reactions to happen inside of the cell. Nutrients provide the energy for life-sustaining chemical reactions, but they also provide the raw materials that allow microbes to build proteins, lipids, nucleic acids, and carbohydrates, which are essential for cellular structures. Microorganisms survive at specific pH ranges because the enzymes in their cells will denature at high pH or low pH at the extremes. So it's up to scientists to provide buffers that inhibit large pH changes in microbial environments. Culture media is produced in the laboratory to provide the water, the nutrients, and a pH-stable environment for microbial growth. A culture medium in which the exact chemical composition is known is called chemically defined, and it's exemplified by glucose minimal salts, which is the recipe on the left. Notice that in this type of medium, every ingredient has a specific chemical formula, so we know exactly how much of each kind of nutrient or element is in the medium. This means that each time we make the medium, we can be assured that as long as we make the correct measurements, the medium will be consistent and the same each time. Usually, scientists will use chemically defined media when they want to know something specific about the metabolism of a specific microbe. Most bacteria are routinely cultured on complex media. So complex media is what you see on the right. Nutrient agar is an example of a complex medium. This is a medium for which the exact composition varies from batch to batch. You'll be able to recognize this type of medium when you see it because the ingredients will include words like extract or peptone. Peptones are made when cells are lysed and their proteins are partially digested, as is the case of, again, your peptones. Or in the case of extracts, maybe a cell is broken open, the proteins are extracted, and then they're powdered. So that's what happens with yeast extract. Beef extract is made when a big piece of beef is boiled in water, and then that material, that broth, gets dehydrated. The point is that for the yeast extract, the peptones, or the beef extract, none of the processes for making those ingredients is very precise. So that means that the composition of these ingredients can vary. And then when that ingredient is included in broth or in a growth medium, it means that the composition of that medium may also vary from batch to batch. The goal of a complex medium is usually to grow as many microbes as possible, so differences from batch to batch aren't really that important. After the ingredients for culture media have been mixed and everything is dissolved, the medium is then poured into glass bottles or test tubes and it's sterilized. The most common method of sterilizing media is to place it in an autoclave. During autoclaving, the medium is treated with steam at a pressure of 15 psi, that's pounds per square inch. The pressure raises the temperature of the steam from 100 degrees Celsius to 121 degrees Celsius, and then the medium is held at 121 degrees Celsius for at least 15 minutes to kill all forms of microbial life that may be in the medium. You'll want to remember 121 degrees Celsius and 15 psi for a minimum of 15 minutes to sterilize. You could see that information again, hint, hint. After the medium is cooled, it can be inoculated with microorganisms. A sterile broth is almost always clear and transparent, like these examples on this slide. When microbes are growing in the medium, it takes on a cloudy appearance, which we call turbid. Turbid is spelled Turbid. Scientists and technicians use broths to grow microbes to high density. That means we're going to grow lots of cells in a very small space. Microbes in a broth can then be stored for 3 to 4 weeks, but the viability, that is the survivability of the cells, tends to decrease when you get closer to 4 weeks. If a broth contains a mixture of different bacteria, you wouldn't be able to tell. Usually, broths containing mixed bacteria have kind of the same turbid appearance as a broth containing only one type of bacterium. The only way to separate the microbes in a broth would be to do a series of dilutions where you gradually and systematically reduce the concentration of microbes in that broth. And this is a time-consuming prospect. It requires a lot of materials to accomplish, and there are easier ways to do this. Agar is a polysaccharide that's extracted from marine red algae. When you buy it, it comes as a white powder that can be added to broths like the ones I've described. Heat must be applied to melt the agar so that it will mix with the broth. Then the mixture gets autoclaved for sterilization. Once sterile, the agar can be left at room temperature to cool to about 40 degrees Celsius, and then it can be poured into a plastic dish like this one, which is called a petri dish. Or the agar can be poured into a test tube to make an agar slant or an agar deep. Agar culture media provides a surface, a solid surface, for spreading bacteria out and growing them in the lab. A turbid broth could contain one type of microbe or many types of microbes. What we could do, instead of making all those dilutions that I mentioned on the last slide, is we can take a tool like this one, which is called an inoculating loop, and then we can use it to spread the bacteria out on the surface of our agar, like this. Going all the way down the plate, by the way. Then we could incubate this plate at the appropriate growth temperature, and after incubation, we might see that our plate looks like this. Each circular growth that you see here, these small ones like this, as well as the large ones like this, and these over here, these are called colonies or colony-forming units, and each one represents one bacterium or a short chain of bacteria that grew thousands of times to form a clump. As you'll find out later in the lab, Different types of bacteria make different colonies, and we can use the appearance of a colony to help us identify the bacteria. So the power of agar in a petri dish is that it allows us to spread microorganisms out, separate them, and then study them. Agar petri dishes are also useful because agar is not digested by most bacteria. Therefore, it remains solid when we grow all different types of bacteria on it. Also, agar is solid at temperatures up to about 100 degrees Celsius. So that means that we could grow heat-loving bacteria on this medium. Typically, agar petri dishes can be stored for 2 to 3 weeks, sometimes up to 4, but by then you might start growing unwanted microbes or the medium starts drying out. Agar deeps are made when molten agar is poured into a test tube and allowed to cool as the test tube stands upright in a test tube rack. Deeps look like this and are useful for determining the oxygen requirements of a microorganism, particularly when a chemical like thioglycolate is added. Thioglycolate binds oxygen, so that means that as you get deeper into the tube, the amount of oxygen goes down. And some microbes actually prefer low oxygen environments. We can also use deeps for studying motility. To study motility, we would take this. This is called an inoculating needle. It looks like a pin on a handle. We would heat it in a Bunsen burner to sterilize it. Then we'd allow it to cool, dip it into a bacterial broth. Then we would basically stab the needle into the medium in this direction and pull the needle back out in the same path. As microbes grow in this path, if they are motile, that is, they move, they'll actually start to swim away from the path of inoculation. If bacteria are non-motile, they'll stay within that path. So it's very easy to determine whether a bacterium you're working with moves or doesn't. We can also observe gas production because if microbes that are growing within the stab line here produce gas, then we might see the formation of little bubbles off to the side. Agar slants are made when molten agar is poured into a test tube, but the test tube is laid at an angle. So the agar cools, making a slanted surface, which you can see right here. Typically, this type of agar is used to store bacteria longer term, for about 4 to 5 weeks. It has a reduced risk of contamination because the only way that bacteria can get into the tube is through this opening, which if the cap is on there, the microbes can't come in. This medium also doesn't dry out as quickly as a petri dish. Remember, this is a petri dish. The slant doesn't dry out as quickly because there's only a small surface area and only a small amount of air here in the tube. So that means that water is less likely to diffuse out and dry out. Other equipment that's used in the microbiology lab, this is another view of our tool. This is the inoculating loop. You can tell it has a little loop on the end, which is why it's called the inoculating loop. We use this tool to transfer microorganisms from one culture medium to another. This is called an inoculating needle. It's also used for transferring microbes, but to agar deeps. So what you would do is something called a stab inoculation, where you would stab the agar and the bacteria would grow in the agar along the path that you stabbed. This is a Bunsen burner, B-U-N-S-E-N, burner. It's used for producing flames in the microbiology lab. We use the flame to sterilize implements prior to their use. And you'll probably recognize that this is the striker that we use for lighting the Bunsen burner. What other things might microorganisms need to grow? We looked at water, we looked at nutrients, we looked at proper pH. What else? Proper temperature and oxygen. Now, oxygen has a question mark beside it because in the world of microorganisms, not all microbes actually need oxygen in order to survive. There are some microbes that are actually killed or growth inhibited by the presence of oxygen. Temperature, however, is important for all microbes. I don't know about you, but the last time I looked at bacteria under the microscope, they didn't wear little fur coats because it was too cold, and they certainly weren't wearing t-shirts because it was too warm. If we want microbes to grow, we have to provide them with the proper temperature that will allow their growth. The way that we provide proper growth temperatures for our microorganisms in the lab is by using these. These are incubators. They are temperature-controlled chambers that have little heating elements in them. And the heating elements allow us to raise the temperature above room temperature. So at Bristol, we typically grow our microbes at 37 degrees Celsius. Do you remember what the significance of 37 degrees Celsius is? Right, it's body temperature. And then we'll also have incubators that are set at 30 degrees Celsius. Sometimes incubators will be outfitted to deliver gases to the temperature-controlled chambers, for example, carbon dioxide. Most of our bacteria grow using oxygen and don't require other gases, so most of our incubators actually won't be outfitted for delivering gas. For those bacteria that need oxygen, we simply grow them in petri dishes, and the assumption is that the top of the petri dish is so loose, oxygen can get up and into the medium for the bacteria. And for our test tubes, we usually leave the caps kind of loose so that they jiggle a little bit, but they're not, you can't pull them off. This also allows enough oxygen to get into the tube to support the growth of our microbes. You can't have a micro lab without having a microscope and stains. Microscopes are important because they magnify the appearance of microbes so that we can actually see what the cells look like, if they're making any special arrangements, what their shapes are. If we've stained the cells, we can see what color they are, and that'll tell us something also about the structure. So microscopes are incredibly important. Chemical stains, like the ones you see over here, are important because they add color to microorganisms. Most microbes are actually colorless, and you wouldn't be able to see them under the microscope. But when you add chemical dyes like these, you can start to observe something about microscopic structures. Every lab has to have a place where you dispose of contaminated materials, be they gloves, lab coats, used Petri plates, or test tubes. And this is the one in the Attleboro campus. At Bristol, you'll always know where to dispose of microbially contaminated materials because you'll see this orange biohazard tape surrounding the area that you can place those materials. You'll also see signs like this, which tell you that these containers contain biohazards. Usually we have one container like this one for gloves, and then we'll have another container for things like Petri plates. Slides and pipettes would go in little containers like these. And then test tubes would go into a rack. But the important thing is that all of this is kept in the biohazard area of the lab. This reduces contamination to other areas. Well, that brings us to the end of this video. What I hope you'll take away from it, because it could be on your next lab homework or lab exam, hint hint, are the names of the pieces of equipment and tools in the microbe lab, plus their uses. For example, if I asked you about stains, you should be able to say in your own words that they add color to microbial cells and allow us to examine their shapes, arrangements, and structures. If I showed you a picture of a Bunsen burner, you should be able to tell me that its flame is used to sterilize tools like inoculating loops. I also mentioned some new vocabulary words. In case you missed them, I'll repeat them here. We talked about aseptic technique, which we said were the steps and procedures that help prevent contamination of ourselves, our experiments, and our environment with unwanted microbes. We also mentioned the word culture. So culture means that we're going to grow microorganisms in media in the lab. We culture bacteria in culture medium, which is a liquid, semi-solid, or solid solution containing essential nutrients for microbial growth. Inoculation means that we're introducing microorganisms into culture media where they can grow and reproduce. Sterile refers to the absence of microbial growth. An autoclave is a laboratory device that applies steam and pressure to sterilize. And turbid, well, it means cloudy and indicates the presence of microbial growth. There was a lot of information in this video. Take the time to really get familiar with the tools, equipment, and terms of microbiology. Your investment will make the rest of your class much easier. For now, I'll leave you to it. Oh, and don't worry, not all of your lab videos will be this long.